So hi, my name is Serena. I'm a PhD student in the lab of Caetano Gonzalez, and I will be telling you about our newest findings using a fly model to study a disease called Ewing Sarcoma. So Ewing Sarcoma is a cancer that arises in the bone and soft tissue. It happens to be the second most common pediatric bone cancer, and the current standard of treatment is limited to surgery, chemotherapy, and radiotherapy. There's no specific treatment for this disease as of yet. This is what motivates a lot of the research effort into developing an animal model that will allow studying the disease. But despite extensive work being put in by multiple laboratories, the effort has been largely unsuccessful. This is because the driver of this disease, an oncogene called EWS-FLI, is so highly toxic that the large majority of cell types will die when you try to express a EWS-FLI in them. That is the lack of such models is why back in 2019, our lab decided to try and develop such a model in the fly, in Drosophila. And this was initiated in tight collaboration with Dr. Jaume Mora from the Sant Joan de Déu Children's Hospital in Barcelona. Our initial work showed that, just as in other cell types and in animal models, EWS-FLI is highly toxic in the fly as well. This initially prevented us from obtaining a transgenic fly line that would express EWS-FLI. And our current publication in Open Biology is about how we circumvented the problem of toxicity and utilized various different strategies, as a result of which we have various models expressing EWS-FLI to varying degrees or different levels. So using these models, we were able to investigate the changes in some of EWS-FLI's key functions, and we found that different functions follow different kinetics with respect to EWS-FLI protein levels. Why is this important? Because from studies of human Ewing sarcoma tumors, we know that EWS-FLI levels are highly variable within cells of the same tumor. And we also know that these different levels correspond to highly different cellular responses. We believe that our tools can help to study the consequences of changing and varying EWS-FLI levels, such as those observed in human tumors, and contribute to the search for more specific therapies.